I'm very happy to introduce my brother Atul Jadhav. He is a student of natural products and is working as general manager for Himalaya Global Research Center. He is part of a team that develops products covering dietary supplements, personal care, and animal wellness for GCC and other regions. He holds bachelor's in pharmacy from University of Pune, master's and Ph.D. from in natural products from NIPER Mohali. He has completed his post-doctoral studies at NCNPR, University of Mississippi, and has worked in industrial research labs prior to joining Himalaya. I invite Atul to give his talk.
Thank you for the kind invitation. It's a privilege to interact with students of Pilani. What I'm going to share today here is some piece of work which we have done at Himalaya and overall view on the quality control of botanicals, practice and challenges. So what I intend to cover is like what exactly we are into the business of, why do we standardize, what are the herbal drug processing steps and the general standardization practices, and we will discuss 2 case studies which we have experienced. So as we know, we have been blessed to have a great heritage where we have a traditional medicinal system of Ayurveda and also Siddha medicines. So with this, we have an elaborate knowledge about the usage and processing about the herbs coming from Mother Nature. We are also blessed to have a 3 million square kilometers of area where we have diverse environmental conditions, having different herbs growing in different regions. And so we have a different or a varied biodiversity available to us. So what we usually try to do is trying to understand Ayurveda and having it contemporized. So as we understand, Ayurveda is the science of life, and it is one of the oldest healing systems in the world with a recorded or documentation going back as long as 5,000 years back. This system is not fractionist like what the modern medicine actually works like. It tries to treat a particular patient with a holistic approach, and a primarily use of the treatment regime is botanicals. It's not only preventative, it's not only curative, it's also preventive. So it actually makes a difference there with respect to the current practices, what we see. So at Himalaya, we have a big spectrum which we address, which is for dietary supplements of what we call Ayurvedic proprietary medicines in India. We have personal care products, we have animal health products wherein we work with botanicals. Himalaya had a very humble start, which dates back to 1930s, where one man had a vision to bring traditional science of Ayurveda to the world in a contemporary form. Now, when we recollect these periods, this was the periods where we were seeing penicillins coming into the force with a modern allopathy taking a bigger shape. And this man was against the tide. We can understand that how could be how tough that journey could have been. Currently, Himalaya is present in more than 89 countries. Our global headquarters is at Dubai, where currently we are also having an R&D center. Our products are prescribed by more than 4 lakh doctors worldwide. We have the trusted millions of customers globally. So when we talk about herbal drugs, we say that why do we need a standardization practice for it? So as we understand what our core business will be, having Ayurvedic proprietary medicines developed from the science of Ayurveda. So basically contemporizing the traditional medicine to international standards. So it will need a lot of scrutiny, and we need to deliver efficacy and safety each time, batch after batch. So it will mean that you need to standardize something which is natural, which has its own intricacies involved, and it's supposed to have its own diversity. Typical modern drug discovery from herbs has a fractionist approach where the herbal extracts are done, they are subjected to different in vitro, in vivo screens. Then there will be a fractionation, and then keeping on the fractionations against the screen, you get a heat. So this approach versus the approach what we use is quite different. But the practices, what basically we look at is we need a quality to be sure of the documentation needs of it, the standardization part of it, and the clinical evidence of it. So these four principles anyway again apply to herbal drug development, but while doing so, because of its own innate nature, it gives certain challenges. For example, there could be a wrong plant species for an intended pharmacology. You may choose a right plant, but a toxic one. So you need to have a processing for that. You may choose a related species, may not be exact species, and then you will land up with an inferior quality. There could be incidental contaminants because it is going open in air, and with the soil, the contaminants like heavy metals can come, the pesticide residues can come, and the microbes will always be there. There could be deliberate adulteration for commercial purposes, or there could be substitutions or some extraneous matters added into that. So this all is going to affect the herbal drug quality. Then there could be foreign matters. Then in the processing as well, there could be a substandard processing which is used. And some of the times there is a poor information or evidence on the safety and effectiveness. So these are the risk areas when you venture into herbal drug development. What determines botanical quality? So basically what we rely heavily on authentication, which is traditional pharmacognosy, what we know in pharmacy schools. Is it a tool? Yes, it's a great tool. Even today in industrial practices, we always use these tools because there will be species which are interrelated. Unless until you do pharmacognosy, you will not be able to threadbare understand which species you are handling. Purity, limits of foreign matter. So in this raw materials, when it comes in gummy bags, you will be seeing that there could be certain extraneous matters coming in it, or non-useful parts of the plant, or some adulterants also. So you need to be very sure whether it is free from stones or some other thing. So you need to set up some limits for that as well. Then it will need, since the results will be, or the efficacy will be mediated through certain chemicals, we need to have a chemical standards and their analysis. And this poses a big challenge because there may not be every time you have some standard known for a particular plant. You can also choose to do a chemical or biological assays, and then the packing, labeling, and storage conditions also affect the botanical quality. Why do we standardize? Basically, in this case, we need to deliver desired efficacy each time, every time. In modern medicines, API is precisely controlled. So you have an assay, say for 99.9%, you know even the impurity is 0.1%, what is the impurity there. Now in this case, our API is herbal extract, and we need to maintain the therapeutic window with these uncertainties around it. But we need to achieve a similar precision. With the herbal extracts, we have a lot of challenges where we have a innate nature where there are dozens of chemicals, related chemicals, diverse chemicals which are associated. It also gives a geographical and seasonal variation. So if you notice, there will be a different bilanolide content across if you harvest it in different conditions. In physics, there could be a physical chemical contaminations because of the commercial supplies. For example, if you have to get Postvalia gum, you will see it loaded with a lot of stones. So it's an innate nature because the dust will go and sit with the gum. All of this also gives challenges for us, and that's why we need to have standardization effect. So it is important that we are checking that the materials we are handling are not harmful in any way, and there is also quality, purity, and stability known for these materials. So standardization of herbal substances and preparation, basically, as we briefly said, it's depending on the botanical source, part use and its state, whether it's a whole drug or it's a powdered material. Then the characteristics like organoleptic, microscopic are also important. So these identifications stages of microscopical and macroscopical characters are still used. The chromatography and chemical reactions are heavily used. In the test, the foreign matter, total ash insoluble in hydrochloric acid, water-soluble extractives, extractable matter. This seems to be academic interest, but no, these are also industrially useful because you can sometimes control the extracts where there are not much known about the chemistry of it. We also need to know the contaminants because we need to limit those heavy metals and microbial stuff or mycotoxins onto the product. And as I said, heavily wherever the markers are known, the active markers or reference markers are known, we need to have an assay of those. What are the problems associated with standardization of herbal preparations? So as we understood that there are a couple of challenges which naturally comes. Moreover, from the science perspective, we don't have a complete understanding of the exact mechanism of action, how this herbs will be 100% acting. There are a couple of dozen components which are going inside with the herb and which and all receptors are involved is not completely known. The literature evidences for biomarker compounds, sometimes, for example, Cissus, are known, but they could be questioned. Then whatever biomarker you are going to select, whether it's a correct biomarker or there could be an alternative biomarker. If you don't have a biomarker, which analytical marker do you want to choose? Will it give a characteristic for that particular material? These are all the questions we have. Then when you actually venture into a standardization with a marker, there could be a challenges of commercial availability of such marker compounds. Then we have a problem with availability of fast and economic methods of determination. So because the quicker, the better. It saves time and money. But there are no standard practices for this as on today available. When a complicated herbal formulas are there, say for example, you are having a 7-8 herbs together, it actually gives a challenge with respect to a large pool of chemistry there. And controlling quality in this gives a different challenge to a formulator. Moreover, on top of it, there is no uniform regulations. So certain places, certain herbs are banned or heavy metal limits are changed in different geographies. So all these are challenges for you to have a global product. So when we look for standardization, we have two aspects here. One is a herbal drug substance or preparations coming out of it. So basically, when you have a powder material, you need to understand the particle size, the nature of the material, whether it is thermally sensitive because the powdering process will generate heat and whether it is going to affect the chemistry there, we need to understand it. Also, the people who are handling, is there any conditions which may occur while handling like allergies or something that we need to be careful. Then we need to have an extraction plan for that. Either it would be a single herb extraction or a multitude of herbs together getting extracted. You need to choose medium of extraction that is suitable for your purposes. And you also need to optimize the process parameters to get a maximum yield out of it. If yield varies even with 1%, your commerce calculation changes. So every small aspect of this is important for the extraction processes. When a particular extract is prepared, we need to see the finished dosage form, how it is getting formulated, whether it's a liquid product or it's a solid dosage form. And that will have its own limitations to handle the materials into the final formula. So what formulation development requirements are there needs to be mapped while developing an extract. We also can use liquid concentrates which are easiest way out, or there could be drying, and in drying there are several techniques of drying. So that also plays a role with the cost and the quality. So when we say about the extraction, as we said that there is multitude of chemical components there in a herb, there could be a different choice of solvents like non-polar, medium polar, or polar. And then the marker components will definitely be getting affected. So for example, we are seeing Borvia diffusa here, which is extracted in ethanol, hydroethanol, water, and hydroethanol ratio. So if we see that the profile coming out of the same herbal material with the same extraction conditions with a different solvents, you can see a range of chemistries getting expressed in a different way. So standardization using marker compounds is a preferred way, as we understand that herbs have a complex chemistry with primary and a secondary metabolites. So which ones we usually choose as a secondary metabolites, which ones to choose? There could be a ubiquitous compounds, there could be a special class of compounds which represent that particular genus. So all that we need to thoroughly thought out. Then we also need to understand if we are having multiple herb combinations, is there synergy there, antagonism there? Do we have any toxicity or safety issues going to be created with that? We need to have bioactivities established. So for example, in Boswellia, boswellic acids are known as a bioactive marker. So you will be needed to choose a proper biomarker as boswellic acids only in this case. If you don't have a choice of this kind of markers, we need to choose something which is a representative of it. For example, you can choose gallic acid for commonly found amla fruits. In absence of any of this kind of markers, you can always choose group markers. Even those are useful industrially. Just to elaborate the importance of all of these standardization parameters, I'm just going through some of the case studies. The first one is Cissus quadrangularis. This is popularly known as Had Jod in Ayurveda, the name Had Jod. So it basically to be Haddi Jodne Wala. So that's how the name Had Jod has derived. So it has not got any well-established marker compounds, and but much of the talk in the literature is about ketosteroids, which is a gravimetric assay. And there in the Western markets, because of the ketosteroid word, there is a traction that it supposedly improves the athletic performance, and there is no validity for this kind of claims, but there are certain notions in certain regions which exist like this. So the commercial people exploit such a situation or such understanding in the market, and there will be products flooded with a claim that we are having a higher ketosteroid assay, intending to suggest on a higher athletic performance activities. When we say that you are having a high ketosteroid content, arguably you can question that there should be a single individual marker, but most of the time there is no answer for such a question. So there was a question with us whether the ketosteroid is a hype thing, is it a truth, are we having a correct information in literature? So with these questions in mind, we started our journey studying Cissus quadrangularis. Going by the classical understanding, it could be an aqueous extract that should be useful. So we had one Cissus quadrangularis extract which was in-house, which is a water extract. We also obtained some samples from western region of the world. So there were certain samples which were standardized, certain with claims like 40% ketosteroids, 20% ketosteroid. We used one ubiquitous marker here in this case, which is beta-sitosterol. We also envisage that if you have an ethyl acetate extract, you should be able to get a higher concentration of ketosteroids into the extract from the herb. So if you see the fifth track is of ethyl acetate extract. So basically what we are trying to track with the ketosteroid in this particular herb is they are non-polar constituents of the herb and would be easily getting extracted into ethyl acetate kind of environment. And in water extract, if you see the track 1, you will see that it's lesser or it's absent in some of the spots or tracks. So the commercial supplies, which are with some assay limits like 40% and 20%, hint at that the commercial supplies may be extracted with organic solvents and not with water. Now, how these assay claims come of 20% ketosterol or 40% ketosterol? There is no harmonious method for that. Certain places there is method A used and method B used. I'll explain what this method A is. Now, in method A, the sample is extracted. That is, the extract is extracted in 80% methanol. Then it is partitioned against diethyl ether, and the ether layer is washed with alkali. The ether layer is again washed with water. It is dried with and sodium metabisulfate. And then the total gravimetric weight is taken, which is expressed as total 3 ketosteroids. With this, if you do a ethyl acetate extract estimation, you get a assay of ketosteroids around 1.4%. Now we have another method D, which is also used in some certain industries. Here the sample is directly extracted in diethyl ether. It is washed with alkali. And this diethyl ether layer is again washed and dried. And you get a gravimetric assay of 12.92. Now here what we see is that if you use method A and method B, you don't get the similar results. There is a 10 times difference between the two results for ketosteroid. So when a commercial setup, somebody wants to say that I'm having a higher ketosteroid assay, there is a good chance that he will prefer method B over method A for obvious reason of having claimed higher percentage of ketosteroid. So in short, we also tried to understand what will be the impact of different solvent processes. So ethyl acetate, supercritical extracts, and aqueous extracts. So all of these were analyzed by method A and method B. So based on this, what we found is that the gravimetric ketosteroid assay is highest with CO2 extract, then ethyl acetate extract, and the lowest is with aqueous extract, which is true because aqueous extract may not be able to get all that ketosteroids extracted completely. So what should be the medium of extraction? So if you get a CO2 extract, you can standardize with beta-citosterol, ethyl acetate extract can be done. But as we notice in this TLC fingerprint, beta-citosterol is obscure in the Hutjod extract, which is aqueous extract. So this is on the chemistry front. How does it correlate to the activity? So one of our pharmacology team subjected these extracts to study their effect on the bone parameters. And as it is evident from this table, you would see that when you are comparing the aqueous extract with the ethyl acetate extract, a CO2 extract, the aqueous extract is surprisingly doing better than these two extraction mediums. Now, Hutjod, when you say that the aqueous extract, this chemistry will definitely be changing and will have a more larger picture than only ketosteroid picture to this particular extract. So from this case study, what we understand is that perhaps in the case of Cissus, where the ketosteroid story is afloat, we do not know the exact component and fraction that is bioactive. And ketosteroids, when the group ketosteroids are seen, we haven't seen any particular marker compound which is characteristic there. In my personal experience, wherever I have studied even using NMR spectroscopy to identify the keto fraction, we have not detected anything useful. But we can conclude one thing safely, that ketosteroid assay does not translate into biological effects. So there is much more beyond to the ketosteroid assay for this. So the current practice of using ketosteroid as an assay can be easily questioned. So this method of diethyl ether fraction standardized extract cannot be construed for an activity or a bone healing or anabolic activity. This work was subsequently published in one of the journals. I would like to take another case study, which is of Tribulus terrestris, Gokshira, and I think this is abundantly present in your areas of Rajasthan. So Tribulus is again portrayed as an aphrodisiac product in Western worlds, whereas it has multitude of different activities. The aphrodisiac activity has been studied in animals and has been shown to be having a good aphrodisiac activity. Further, in literature, it is also established that ferrostenol glycoside fraction is responsible for such an activity. They also showed increased serum testosterone level in male rats after 14 days of treatment. And with this story, in US, there was a surge in the interest for this particular product. Now, with this product in commerce, there started commercial race who can sell better, why my product is better. So the obvious pitching is saying that higher assay is associated with one particular capsule or a tablet. So there started a commercial race where people started saying that higher assay with a higher value for money kind of propositions. So again, in this, when you look at the chemistries, the two varieties are existing, Bulgarian and Indian Tribulus. And Bulgarian Tribulus is supposedly reported, or rather it is reported to be rich in protodioscin and protodiabestine. And supposedly it has a higher concentration, so it is having higher aphrodisiac activities, is the commercial sense with protodioscin assay. Indian Tribulus is known to contain higher concentrations of tribulosine. So both of these chemistries are differing, like one is pyrostenol, one is spirostenol molecule. Now this protodioscin, is it only present in Tribulus? So no, it's also present in Trigonella and Dioscorea. How this protodioscin is distributed in the Tribulus. So there are we basically see two different chemotypes in relation to protodioscin content. One is the segment where you see crop coming from Bulgaria, Turkey, Greece, Serbia, Macedonia, Georgia, Iran, representing one chemotype, and the another one is Vietnamese and Indian. So Vietnamese and Indian show a particular chemotype, and the other ones are Bulgaria. So all of this region of Bulgaria gives a similar profile with protodioscin and prototriabestine to be a major component. In the Indian and Vietnamese chemotype, you will see tribulosine as a major component, and certain places it is claimed that prototriabestine and triabestine are not present in these varieties. And it also has uncharacterized components. So with this, we had a hypothesis with us that if Bulgarian Tribulus is commercially promoted because of the higher protodioscin content and associated higher aphrodisiac activity, is this the correct proposition? So we had thought process to evaluate. Bulgarian Tribulus contains high concentration of protodioscin, and protodioscin is the active compound. So we started with one commercial extract, which was standardized Bulgarian extract, Bulgarian Tribulus extract, where we actually first determined its protodioscin content by HPLC. So it was really high with 8.35% of protodioscin and the lesser concentration of tribulosine, which is 0.54. And as we see in the Tribulus aqueous extract here, which is Indian extract, you see a concentration which is higher of tribulosine, which is at 1.58%. But we also see a presence of protodioscin in the Indian extract. So it's not absent here. So this is one typical assay methodology, which is called as Bulgarian spectrophotometric assay. So we decided to use this methodology to understand even the Indian Tribulus extract, how it fares with this content. So the commercial sample which was used actually showed a very high content of 26.87% of protodioscin translated by spectrophotometric assay. alcoholic extract of Indian Tribulus showed 4.53 and a water extract showed 4.11%. So now with this, you can understand a commercially viable proposition is Bulgarian Tribulus, which is supposedly containing higher protodiocin, which is claimed in literature to be responsible for aphrodisiac activity. So because of this, the Indian Tribulus in the Western markets got affected and the Bulgarian Tribulus dominated the market. So we wanted to understand how these chemistries are different. So if you see a TLC profile here on the rightmost lane, you see that it is the Bulgarian extract, which is actually showing a higher protodiocin content. And the leftmost, the first track on the left is the Indian Tribulus extract. So on a metabolic level, as we say that furostenol, pyrostenol, glycosides are not very structurally distinct molecules. And as we understand the biochemical transformation that may happen within the body, there could be a change. That was the thought. And actually there are some reports which shows that these forms of a particular protodiocin can get converted into spirostenol glycoside as well. So when we have this understanding from literature, so let's try to understand whether protodiocin is a hype or it's a truth that it is the responsible molecule. So as we see here, if you consider the molecule as a two parts, one is a lipophilic part and one is a hydrophilic part, as it's known to us that hydrophilic part is helping for the intestinal transport as the lipophilic part is poorly absorbed. So together this is amphophilic and it gives a emulsification property. So we also understand that there will be intestinal and microbial enzyme background for this molecule. So a spirostenol can become a furostenol as well and vice versa. So when we see these, what we understand from these studies is there could be a geographical variations and a different chemoprofiles. But when a particular assay method is used, you will get a different results with different chemistries. And a claim of protodiocin being the active could be questioned. So it may not be only protodiocin that is the active, but a group of all of these pyrostenol, furostenol molecules could be the active principles. We also studied the aphrodisiac activity of Indian Tribulus and we compared it with the Bulgarian extract. And in fact, again here to our surprise, Indian Tribulus showed better results. So with these two case studies and overall the process of standardization, what we understand is that standardization is a key for safe and efficacious herbal drug. It involves multifaceted activity of a compound and also multi-component activities like synergism, antagonism, etc. Holistic herbal drug development uses established traditional indications and doesn't go with a particular hype for a particular indication. The holistic drug development poses many challenges to phytochemists, wherein the efficacy studies are not correlated for particular chemical components or chemical markers are not completely known. And when we study this actually opens a lot of questions, and I would invite all of you, the brighter minds, to join us in studying this because it's a Pandora box, and for all motivated biologists and phytochemists, it's an open area. With this, I would like to thank all our internal teams, analytical development team, pharmacologic team, and discovery sciences team who have contributed for this piece of work. And thank you for your patient hearing.
Atish has a question, Atul. He has written in the chat box.
Okay, there's a question from Atish. Are quadrangularis, stilbene derivatives, the active phytoconstituents in Cissus quadrangularis in addition to ketosteroid? Have they been investigated at Himalaya? Frankly, Atish, this is not yet evaluated at us. The piece of work which I just now presented is a bit old, which we published in 2016 or 17 around. and still beans could be active. So considering that something is active and something inactive is a very bold step. So we should actually look at the herb and the herbal extracts as a holistic approach. But yes, that's a point. I think if given a chance, I would request your team to investigate this still bean part at your lab and publish it for the larger benefit of the community.
Well, I have one question. See,
Yes.
generally in Ayurveda we have either alcoholic or aqueous extract, but most of the components which are proposed in literature are isolated from organic, except alcohol, organic solvent extracts. So, the Himalaya take those organic extracts also in the preparations, or they go by only traditional methods?
To answer it, I will give it in two phases. One is to add to your first statement. Ayurveda has a very elaborate process of extract preparations. There are oil preparations, there are milk preparations, there are ghee preparations. So if we start correlating that, it will actually correlate to the secondary metabolites, which we usually as a phytochemist look at through organic solvent extractions or alcohol-based solvent extractions. In addition to that, answering your second part, yes, we look into all of these aspects when we are developing, and we use modern tools of carbon dioxide extractions in a bigger ways also. So we use a different tool. Some of our products contain CO2 extracts, a water extract, even a whole herb powder also. So the overall result is achieved through a meticulous understanding. And just for the records, although herbal drug discovery seems to be an easy journey for many people, for us at Himalaya, it takes actually 6 years to 8 years to get one pharmaceutical product out into the market. And it's a very rigorous journey with a lot of scrutiny happening into it.
Thank you. So is there any other question? Anyone has any question, you can ask. Fine. Thank you, Atul. Thank you for nice presentation.
Thank you so much for you giving this opportunity, and happy to be associated with the students of BITS Pilani. I wish all the students a great success ahead, and anything we can be of help, please feel free to reach out. Thank you.
Thank you, Atul. Thank you.